As you settle into rest, it can be comforting to know that even as the body grows still, a quiet world within you remains gently active. Beneath the surface of awareness, countless processes continue in a steady, unhurried rhythm, sustaining balance without asking for attention. Digestion is one of these processes, unfolding calmly from moment to moment, guided not only by your own cells, but also by a vast community of microscopic life that shares your inner space. The human body is often described as a single organism, yet in reality it is more like a shared landscape. Trillions of human cells, each with their own roles and rhythms, coexist within a carefully maintained internal environment. Alongside them live trillions of microorganisms, bacteria, archaea, fungi, and other microscopic forms of life that have adapted over long stretches of evolutionary time to live within us. This living collection is known as the human microbiome, and it is not an intruder or an invader, but a quiet partner in daily biological life. Most of these microorganisms reside within the digestive tract, particularly in the winding passages of the intestines. Here, warmth, moisture, and a steady supply of nutrients create a stable habitat. For microbes, the human gut is a place of shelter and continuity. For the human body, their presence offers assistance, balance, and subtle biological support. This relationship is not hurried and not dramatic. It is the result of gradual cooperation, refined over thousands of generations. Digestion begins long before food reaches the stomach. Even the act of eating initiates a coordinated sequence of movements and chemical signals. Muscles contract gently, glands release enzymes, and nerves quietly relay information between organs. These actions are familiar and largely automatic, requiring no conscious effort. As food progresses through the digestive tract, it is gradually transformed, broken down into smaller components that the body can absorb and use. Yet human digestion alone cannot complete this task. Many substances found in food, particularly complex carbohydrates and fibers, resist breakdown by human enzymes. These materials move through the upper digestive tract, mostly unchanged, eventually arriving in the lower intestines. It is here that the microbiome becomes especially active. Microbes possess metabolic abilities that human cells do not, allowing them to gently dismantle these complex molecules into simpler forms. This process unfolds slowly and quietly. Microbial digestion is not forceful or abrupt. It takes place over hours, guided by chemical gradients and gradual growth. As microbes break down fiber and other compounds, they release byproducts that nourish the cells, lining the intestine, and support metabolic balance throughout the body. These substances are absorbed, transported, and put to use without fanfare, contributing to the maintenance of internal stability. The idea that the body contains so much non-human life may at first seem surprising, yet it is entirely natural. From an evolutionary perspective, humans and microbes have developed together, shaping one another's survival. The immune system, for instance, is not designed to eliminate all microbes. Instead, it learns to distinguish between those that belong and those that pose a threat. This ability develops early in life and adjusts gradually over time. The digestive tract itself is structured to support this balance. Its inner surface is not a simple tube, but a living interface, folded and layered to increase surface area and regulate exchange. Cells here are constantly renewed, forming a flexible boundary that allows nutrients to pass while keeping most microbes at a respectful distance. The microbiome, in turn, helps reinforce this boundary by occupying space and resources that might otherwise be taken by less beneficial organisms. What makes this system remarkable is not only its complexity, but its calm reliability. You do not need to instruct your digestive organs when to act, nor guide your microbiome in its daily work. Each part responds to local conditions, adjusting gently to changes in diet, routine, and environment. When changes occur slowly, the system adapts. When rhythms are steady, balance is maintained. During rest, digestion continues at an unhurried pace. Muscular contractions guide material forward. Blood flow supports absorption and transport. Microbes continue their quiet activity, dividing, metabolizing, and interacting with their surroundings. In fact, many restorative processes function most efficiently when the body is at rest. Thinking of digestion and the microbiome as an internal ecology can help place these processes into perspective. Like a forest or a coral reef, the gut environment is shaped by relationships rather than by any single dominant force. Diversity supports stability. Cooperation reduces waste. Gradual change allows resilience. No single organism, human or microbial, controls the system alone. As breathing slows and muscles soften, awareness drifts inward. Meanwhile, this internal ecosystem continues to function, responding not to conscious thought but to chemistry, rhythm, and time. The food you have eaten, the microbes that reside within you, and the cells that line your digestive tract all participate in a shared process that has sustained human life for countless generations. In the hours ahead, as sleep deepens, digestion will move into quieter phases. Some processes will slow, others will continue steadily. The microbiome remains present, patient, and persistent, adapting gently to the conditions provided. Nothing needs to be forced. Nothing needs to be hurried. The body knows how to care for itself, drawing on the collective work of its many living parts. This is the world within you, active yet calm, complex yet balanced, familiar yet often unseen. In understanding it, there is no need for effort or analysis, only quiet acknowledgement. As you rest, this inner partnership continues, sustaining you softly, moment by moment, through the night. 2. The body as an ecosystem. When we think of ecosystems, the mind often drifts outward to forests, oceans, or grasslands, places where many forms of life coexist, each influencing the others in subtle and ongoing ways. Yet an ecosystem does not require open sky or distant horizons. One of the most intricate ecosystems you will ever inhabit exists quietly within your own body. It is enclosed, warm, and stable, shaped not by weather or seasons, but by chemistry, rhythm, and time. An ecosystem, in its simplest sense, is a network of living organisms interacting with one another and with their physical environment. Within the human body, this definition applies with remarkable accuracy. Human cells form the foundational landscape, building tissues, organs, and systems that regulate temperature, fluid balance, and nutrient flow. Within this environment live vast populations of microorganisms, each responding to the conditions provided, each influencing the whole in small but meaningful ways. Unlike external ecosystems, the body's internal environment is carefully regulated. Temperature remains within a narrow range. Acidity is monitored and adjusted. Oxygen, water, and nutrients are distributed continuously. These stable conditions allow life to flourish at microscopic scales. For microbes, the body is not a hostile place, but a dependable habitat shaped by predictable patterns of eating, resting, and activity. Within this internal ecosystem, no single organism acts alone. Human cells rely on microbes to help complete certain metabolic tasks, while microbes depend on the body for nourishment and shelter. This interdependence is not conscious or intentional. It arises naturally from shared survival over evolutionary time. Those microbial species that could coexist without harming their host were more likely to persist. Likewise, human physiology gradually adapted to their presence, incorporating microbial activity into normal function. Balance is the defining feature of a healthy ecosystem. In nature, balance does not mean stillness, but dynamic stability. a state where change occurs gradually and disturbances are absorbed without collapse. The same principle applies within the body. Microbial populations shift subtly from day to day. Some species increase, others decrease. Yet overall function remains steady, supported by diversity and redundancy. Diversity within an ecosystem provides resilience. When many different organisms perform overlapping roles, the system can continue functioning even if conditions change. In the gut, hundreds of microbial species may participate in digestion, nutrient processing, and environmental regulation. If one population declines, others often fill similar roles, maintaining continuity. This quiet adaptability helps explain why the digestive system can tolerate variation in diet and routine without immediate disruption. The physical structure of the digestive tract contributes to this ecological balance. Different regions provide distinct environments, shaped by acidity, oxygen, availability, and nutrient content. These conditions determine which microbes can thrive in each area. Just as certain plants grow only in specific climates, microbial species occupy niches suited to their metabolic needs. Together, these niches form a layered ecosystem, extending from the stomach to the colon. Energy flows through this system in predictable ways. Food enters as raw material, containing chemical energy stored in complex molecules. Human enzymes begin the process of breaking these molecules down, extracting what they can. Microbes continue the work, transforming compounds that would otherwise pass unused. Waste products are recycled or eliminated, and energy is conserved rather than wasted. This efficient exchange mirrors the flow of energy through larger ecosystems, where little is truly lost. The immune system functions within this ecosystem not as a constant enforcer, but as a regulator. Rather than eliminating all microbial life, it monitors population behavior, responding when balance is threatened. In this way, immunity resembles environmental regulation in nature, where predators, resources, and space limit unchecked growth. The goal is not sterility, but harmony. Communication within this internal ecosystem occurs through chemical signals rather than sound or sight. Cells release molecules that convey information about availability, stress, or abundance. Microbes respond to these signals, adjusting their activity accordingly. Human cells, in turn, respond to microbial byproducts, altering gene expression and metabolic pathways. This ongoing exchange forms a feedback loop, allowing the system to self-regulate without conscious control. Time plays a central role in maintaining ecosystem stability. Rapid, extreme changes can overwhelm regulatory systems, while gradual shifts allow adaptation. Within the body, patterns of eating, sleeping, and movement establish rhythms that microbes learn to anticipate. When these rhythms are consistent, microbial populations synchronize their activity, contributing to efficient digestion and steady energy flow. When patterns change slowly, the ecosystem adjusts with them. Rest is an important phase in this cycle. During periods of reduced activity, the body reallocates resources toward maintenance and repair. Digestive processes continue, but at a slower pace. Microbial metabolism remains active, quietly processing remaining nutrients. This steady background work supports cellular renewal and prepares the system for the next period of activity. In this sense, rest is not an absence of function, but a different mode of operation. It can be reassuring to recognize that this ecosystem does not require management or oversight. You do not need to supervise digestion or guide microbial behavior. These processes are governed by physical laws and biological feedback mechanisms refined over immense spans of time. The body's role is to provide a stable environment. The microbe's role is to respond to it. Together, they maintain equilibrium. Seeing the body as an ecosystem also shifts how we understand health. Health is not the elimination of all change or variation, but the capacity to absorb change without losing balance. Just as a healthy forest can withstand storms, a healthy internal ecosystem can adapt to variation in diet, environment, and routine. Stability emerges not from rigidity, but from flexibility supported by diversity. As you continue to rest, this internal ecosystem remains quietly active. Cells exchange nutrients, microbes metabolize compounds, signals pass back and forth across microscopic distances. None of this requires effort or attention. It proceeds according to patterns older than conscious thought, guided by the same principles that govern life everywhere. Within you, an entire world continues its gentle work. It is not separate from you, nor is it something to control. It is part of what you are, a living system composed of many lives, moving together in balance. As sleep deepens, this balance holds, steady and reliable, carrying you calmly through the night. 3. Defining the human microbiome. The word microbiome may sound technical at first, yet the idea it represents is both simple and deeply familiar to life on Earth. At its core, the human microbiome refers to the full collection of microscopic organisms that live on and within the human body, along with the environment they inhabit and the activities they perform. These organisms are small beyond ordinary perception, yet together they form a presence so vast that they rival the number of human cells themselves. Most members of the microbiome are bacteria, single-celled organisms that have existed on this planet for billions of years. Long before humans appeared, bacteria were already shaping Earth's atmosphere, oceans, and soils. Over time, some of these ancient lineages found stable refuge within larger organisms, including humans. In this protected environment, they adapted to new roles, no longer living independently in open ecosystems, but as residents of a shared biological space. The microbiome is not limited to one location. Microorganisms live on the skin, in the mouth, along the respiratory tract, and throughout the digestive system. Each of these regions offers distinct conditions, and each supports different microbial communities. The skin, exposed to air and light, favors organisms adapted to dryness and variation. The mouth, warm and moist, supports others. Yet it is the digestive tract, especially the intestines, that hosts the largest and most diverse microbial population. To understand why, it helps to consider what microbes need in order to survive. They require warmth, moisture, and a steady supply of chemical energy. The human gut provides all three in abundance. Food arrives regularly, broken down into increasingly smaller components as it moves along. Water is present. Temperature remains stable. For microbes, this environment offers continuity and predictability, qualities that support long-term coexistence. The term microbiome includes not only bacteria, but also archaea, fungi, and viruses. Archaea resemble bacteria in size and simplicity, yet they represent a distinct branch of life with unique metabolic abilities. Some archaea in the gut are involved in processing gases produced during digestion, quietly influencing efficiency and balance. Fungi, though present in smaller numbers, contribute to chemical interactions and ecological diversity. Viruses, many of which infect bacteria rather than human cells, help regulate microbial populations by limiting unchecked growth. Importantly, the microbiome is not defined by a single fixed composition. There is no universal list of microbes shared by all humans. Instead, each person carries a unique microbial signature shaped by genetics, early life experiences, environment, and daily habits. Even so, these differences exist within broad patterns. Certain functions are preserved across individuals, even if different microbial species perform them. This functional consistency helps maintain stability despite variation. The microbiome is often described as an organ in its own right, not because it is made of human tissue, but because it performs essential tasks that support survival. These tasks include aiding digestion, producing certain vitamins, influencing immune development, and contributing to metabolic regulation. Unlike traditional organs, however, the microbiome is dynamic. Its composition shifts gradually in response to changes in diet, routine, and environment, always seeking a new balance. From the moment of birth, the microbiome begins to take shape. As an infant encounters the outside world, microbes begin to colonize the body's surfaces. Over time, these early settlers are joined by others introduced through food, contact, and surroundings. This gradual accumulation is not random. The body provides selective pressures that favor organisms able to coexist without causing harm. Those that disrupt balance are usually limited or removed by immune responses and competition from other microbes. The relationship between the microbiome and the human body is best understood as mutualism, a form of cooperation where both sides benefit. Microbes gain access to nutrients and a stable environment. The body gains metabolic assistance and ecological protection. This arrangement does not require trust or intention. It persists simply because it works. Over generations, natural selection favored hosts and microbes that could maintain this balance. One of the most important contributions of the microbiome is its role in processing substances that human cells cannot fully digest. Dietary fiber is a clear example. Humans lack the enzymes needed to break many fibrous compounds apart. Microbes, however, possess a wide range of metabolic tools. Through slow fermentation, they convert fiber into smaller molecules that the body can absorb and use. In this way, what would otherwise pass through unused becomes a source of nourishment. The microbiome also plays a role in shaping the internal environment of the gut itself. By producing acids and other compounds, microbes influence pH levels, which in turn affect which organisms can thrive. This self-regulating system discourages the overgrowth of harmful species while supporting stable communities. It is an example of how ecological principles operate even at microscopic scales. Communication between the microbiome and the body occurs constantly. Microbial byproducts interact with human cells, influencing gene expression and cellular behavior. The immune system monitors microbial activity, responding when balance shifts too far in one direction. Nerves, embedded in the gut wall, relay information to the central nervous system, integrating digestive activity with overall physiological state. None of this requires awareness. It happens quietly, guided by molecular interactions. It is important to note that the microbiome is not inherently fragile. Diversity allows it to recover from change, reestablishing functional balance over time. This resilience is one reason humans can adapt to varied diets and environments across cultures and climates. The microbiome adjusts gradually, learning new patterns as conditions evolve. As you rest, the microbiome continues its steady work. Microbes divide, metabolize, and interact within their shared environment. Their activity is not synchronized to your thoughts or intentions, but to the availability of nutrients and the rhythms of the body itself. During sleep, when external demands are reduced. Defining the human microbiome, then, is not about listing species or memorizing names. It is about recognizing a living system that exists within you, shaped by cooperation, balance, and time. It is a reminder that human biology is not solitary, but communal, not static, but gently adaptive. Within the quiet of rest, this internal partnership continues, sustaining you without interruption. 4. The digestive tract, a guided journey. The digestive tract can be thought of as a long, gently curving passage, designed not for speed, but for patience. It is a continuous pathway through which food is transformed, step by step, into substances the body can use. This journey is not dramatic or hurried. It unfolds gradually, guided by structure, chemistry, and time, with each region contributing in its own quiet way. The process begins in the mouth, where digestion starts almost imperceptibly. Teeth break food into smaller pieces, increasing surface area and making later stages more efficient. Saliva moistens the food and introduces enzymes that begin breaking down certain carbohydrates. This early chemical interaction is subtle, yet important. It prepares food for what lies ahead, setting the rhythm for the entire digestive sequence. From the mouth, food moves into the esophagus, a muscular tube that serves as a calm passage rather than a site of digestion. Here, coordinated waves of contraction guide food downward in a steady motion known as peristalsis. Gravity assists, but it is muscle activity that ensures reliable transport, regardless of posture. This movement is smooth and automatic, continuing even during rest and sleep. The journey then reaches the stomach, a flexible chamber designed for both storage and controlled processing. The stomach's environment is more acidic than any other part of the digestive tract. This acidity helps break down proteins and limits the growth of many microorganisms. Muscular walls gently churn the contents, mixing food with digestive juices and gradually reducing it to a semi-liquid form. This stage takes time, allowing careful preparation before food moves onward. As digestion continues, small amounts of stomach contents are released into the small intestine. This transition is carefully regulated, ensuring that the next stage is not overwhelmed. The small intestine is long and narrow, coiled to fit within the abdomen. Its inner surface is covered with countless folds and tiny projections, greatly increasing the area available for absorption. Here, digestion and absorption occur together in close coordination. Digestive enzymes released from the pancreas, along with bile from the liver, enter the small intestine and continue breaking down fats, proteins, and carbohydrates. These substances are reduced to their simplest forms, small enough to pass through the intestinal lining and into the bloodstream. Nutrients are absorbed steadily, not all at once, and transported to cells throughout the body. The environment of the small intestine is relatively selective. Acidity is lower than in the stomach, and oxygen levels are modest. These conditions limit the number of microbes that can thrive here, though some are present. The primary focus in this region is efficient absorption. Movement remains gentle but continuous, ensuring that food does not linger too long in one place. Eventually, the remaining material reaches the large intestine, also known as the colon. By this point, most easily absorbed nutrients have already been taken up by the body. The large intestine is wider and slower moving, creating a very different environment. Here, microbial life becomes the dominant force. The colon is home to dense and diverse microbial communities, adapted to low oxygen levels and rich in metabolic diversity. These microbes engage in fermentation, breaking down fiber and other compounds into smaller molecules. This process is gradual and produces substances that nourish the cells lining the colon and contribute to overall metabolic balance. Water absorption is another important function of the large intestine. As material moves slowly forward, water is reclaimed, helping maintain fluid balance in the body. This slow pace allows microbial processes to unfold fully, without rush. The result is a careful extraction of remaining value from what was once food, leaving behind only what cannot be used. Throughout this journey, the digestive tract remains in constant communication with the rest of the body. Nerves embedded in its walls monitor movement, pressure, and chemical composition. Signals are sent to adjust muscle contractions and secretions as needed. Hormones released into the bloodstream help coordinate digestion with appetite, energy use, and rest. The structure of the digestive tract supports this coordination. Layers of muscle allow precise control of movement. Glands secrete enzymes and mucus at appropriate times. Blood vessels transport absorbed nutrients efficiently. Immune cells stand watch, maintaining balance between tolerance and protection. Each layer contributes to the overall harmony of the system. Importantly, the digestive tract is not isolated from daily rhythms. Eating patterns influence the timing of activity. Periods of rest allow for maintenance and repair. During sleep, movement slows but does not stop. Gentle waves of contraction continue, guiding contents forward and supporting microbial fermentation. This quiet nighttime activity is part of the body's broader restorative cycle. The digestive journey is not the same every day. It adapts to variation in diet, hydration, and routine. Yet it does so without losing its fundamental structure. The order of events remains consistent. The pace adjusts, but the sequence holds. This reliability allows the body to maintain stability across changing circumstances. Seen as a whole, the digestive tract is not simply a tube, but a carefully organized environment. Each region creates conditions suited to its role, supporting human cells and microbial partners alike. Together, they transform food from something external into something deeply integrated, providing energy, structure, and support for life. As you rest, this journey continues quietly within you. Food consumed earlier moves along its path, guided by gentle motion and chemical balance. Microbes continue their work in the background. Water is absorbed. Nutrients are distributed. Nothing requires attention or effort. The system knows its way. Understanding the digestive tract in this way can bring a sense of calm. It is a reminder that much of life is sustained by processes that function without command, shaped by time and cooperation. As sleep deepens, this inner journey continues, steady and unbroken, carrying the body gently through the night. 5 microbial neighborhoods along the gut. As food moves along the digestive tract, it passes through environments that change gradually rather than abruptly. Each section of the gut offers its own conditions, shaped by chemistry, movement, and available nutrients. These differences create distinct microbial neighborhoods, much like regions within a larger landscape. In each area, certain organisms find the conditions suitable, while others remain scarce. Together, these neighborhoods form a continuous microbial map that extends from the upper digestive tract to the colon. Near the beginning of the digestive journey, microbial life is relatively sparse. The mouth hosts its own communities, accustomed to regular exposure to oxygen and fluctuations in temperature and moisture. These microbes form structured populations on the teeth, tongue, and soft tissues, adapted to a life of constant change. They interact with saliva, food particles, and one another, creating a dynamic yet balanced environment that resets itself many times each day. As material moves beyond the mouth and down the esophagus, microbial presence diminishes. The esophagus is not designed for long-term residence. Food passes through quickly, and the environment does not allow communities to establish themselves easily. This brief passage serves primarily as a connection, ensuring that the transition from one environment to the next remains smooth and uninterrupted. The stomach presents a very different neighborhood. Its acidic conditions make it a challenging place for most microorganisms to survive. The low pH acts as a selective filter, limiting which species can persist. Only a small number of specialized microbes tolerate this environment, and even they exist in relatively low numbers. For most organisms, the stomach is a place to pass through rather than settle. This acidity serves an important role beyond digestion. By limiting microbial survival, the stomach helps regulate which organisms move further along the tract. It reduces the number of microbes that reach the intestines alive, contributing to overall balance. In this way, the stomach functions as both a processing chamber and a checkpoint, shaping the microbial populations downstream. Once food enters the small intestine, conditions become more welcoming. Though still selective, the acidity decreases and nutrients become more available. Yet movement through the small intestine remains relatively brisk, and digestive enzymes continue to break down food components. These factors limit the ability of microbes to form dense populations. Instead, the small intestine supports smaller, more transient communities. Microbes that reside here are adapted to fast-changing conditions. They tolerate fluctuations in nutrient availability and must adhere closely to the intestinal lining to avoid being carried away. Their presence contributes subtly to digestion, interacting with bile acids and other compounds. Still, the small intestine remains primarily a site of absorption rather than fermentation. As material progresses toward the large intestine, the environment changes once again. Movement slows, oxygen becomes scarce, and undigested material accumulates. These conditions are ideal for microbial growth. The colon, in particular, is densely populated, hosting the largest and most diverse microbial communities in the human body. Here, microbes are no longer passing visitors, but long-term residents. In this region, microbial neighborhoods become more complex and stable. Different species occupy specific niches, defined by the types of compounds they can metabolize. Some specialize in breaking down certain fibers, while others consume the byproducts produced by their neighbors. This layered interaction creates a network of cooperation, where the activity of one group supports the survival of another. These microbial relationships resemble those found in larger ecosystems. Waste from one organism becomes nourishment for another. Energy is transferred efficiently, with little loss. This interconnectedness enhances stability, allowing the community to persist even as conditions shift slightly from day to day. The colon's slow pace supports this stability. Material remains long enough for fermentation to occur, giving microbes time to extract energy from complex molecules. In doing so, they produce substances that benefit the host, such as short-chain fatty acids that nourish the cells lining the colon. These substances also help regulate acidity, reinforcing conditions favorable to beneficial microbes. Spatial organization within the colon further shapes microbial neighborhoods. Some microbes reside closer to the intestinal wall, interacting directly with human cells. Others remain suspended within the contents of the gut, forming communities that move slowly along. This spatial arrangement reduces competition and allows multiple populations to coexist without overwhelming one another. The body contributes to maintaining these neighborhoods through constant renewal. Cells lining the gut are replaced regularly, providing fresh surfaces for interaction. Mucus secreted by these cells creates a protective layer, offering a habitat for certain microbes while keeping them at a safe distance. This separation allows cooperation without intrusion, balance without overexposure. Immune activity within the gut also plays a quiet regulatory role. Immune cells sample microbial signals, responding when populations shift too far in one direction. This monitoring does not aim to eliminate microbes, but to preserve balance. In this way, the immune system acts like an ecological steward, supporting diversity while preventing dominance by any single group. What makes these microbial neighborhoods remarkable is their adaptability. Changes in diet alter the types of nutrients available, encouraging some populations to expand while others recede. Over time, the community reorganizes, settling into a new balance. This flexibility allows the gut ecosystem to respond to seasonal foods, cultural diets, and gradual lifestyle changes. Despite this adaptability, the system resists sudden disruption. Diversity provides resilience, ensuring that essential functions continue even if specific species decline. The presence of multiple organisms capable of similar tasks prevents collapse. This redundancy mirrors patterns seen in healthy forests and reefs, where biodiversity underpins long-term stability. As you rest, these microbial neighborhoods remain active. In the quiet hours of sleep, fermentation continues. Water is absorbed, and metabolic byproducts are processed. There is no urgency, no rush. Activity follows established rhythms, guided by availability and need rather than by the clock. Understanding the gut in terms of microbial neighborhoods offers a gentle perspective on digestion. It reveals a system built not on control, but on cooperation. Each region provides what it can, each organism does what it is adapted to do, and together they maintain equilibrium. Within you, these neighborhoods persist, shaped by time and habit, quietly supporting the body through every hour. As sleep deepens, their work continues, steady and reliable, part of the larger harmony that sustains life from moment to moment. 6. How the microbiome begins in early life. Long before memory, language, or conscious awareness take shape, the foundations of the human microbiome are already being laid. This beginning is quiet and gradual, unfolding during the earliest moments of life and continuing steadily through infancy and childhood. It is not a single event, but a slow process of exposure, settlement, and adjustment, guided by biology rather than intention. At birth, the human body transitions from a protected internal environment to the wider world. In this moment, the surfaces of the body encounter microorganisms for the first time in significant numbers. Skin, mouth, and digestive tract begin to receive microbial visitors from the surrounding environment. These early encounters matter not because they are perfect or permanent, but because they initiate a process of biological learning that will continue for years. The newborn digestive tract is structurally complete but biologically inexperienced. Its tissues are ready to function, yet its internal ecosystem has not yet formed. Microbes begin to arrive through contact with caregivers, the environment, and food. Some remain only briefly, while others find the conditions suitable and begin to establish themselves. Over time, patterns emerge, shaped by repeated exposure and selective pressures within the body. In early life, the microbiome is less diverse and more variable than it will be later on. Microbial populations rise and fall quickly, responding to changes in diet and environment. This variability is not a sign of instability, but of openness. The developing gut is exploring possibilities, allowing different organisms to attempt settlement. Through this process, the body learns which microbial partners can coexist without causing harm. Feeding plays a central role in shaping this early ecosystem. The nutrients that enter the digestive tract influence which microbes can thrive. Certain compounds are especially suited to supporting beneficial populations, encouraging their growth while limiting others. This selective nourishment helps guide the microbiome toward configurations that support digestion and immune development. As microbes begin to colonize the gut, they interact with the developing immune system. It learns to tolerate familiar microbial signals while remaining alert to potential threats. This education is gradual and nuanced, allowing the immune system to distinguish between cooperation and danger. The result is not silence, but balance. During infancy, the gut environment continues to change. Some species decline as conditions shift, while others increase. This ongoing reorganization is normal and expected. The microbiome is not striving toward a fixed endpoint, but toward functional stability, a state where essential tasks are reliably performed. Movement through the digestive tract also changes during early life. As the muscles of digestion mature and coordination improves, the timing of transit becomes more regular. This consistency supports microbial communities by creating predictable rhythms. Over time, microbes synchronize their activity to these patterns, enhancing efficiency and reducing waste. The cells lining the gut are actively involved in this process. They secrete mucus, provide attachment sites, and release signaling molecules that influence microbial behavior. These interactions help shape which organisms remain close to the intestinal wall and which remain in the lumen. This spatial organization reduces competition and supports coexistence. As childhood progresses, exposure to a broader environment introduces additional microbial diversity. Contact with soil, plants, animals, and other people expands the pool of potential microbial partners. Not all of these newcomers remain, but each interaction contributes information to the developing system. Over time, the microbiome becomes more complex, resilient, and stable. By later childhood, the microbiome begins to resemble its adult form. Diversity increases, and population shifts become slower and more measured. The ecosystem has learned how to maintain balance, adapting gradually to changes rather than responding abruptly. This maturity reflects not rigidity, but experience a history of successful adaptation. Importantly, this entire process unfolds without conscious guidance. There is no moment when the body decides which microbes to keep or discard. Instead, physical conditions, chemical signals, and biological feedback loops shape outcomes naturally. Microbes that contribute to balance are supported. Those that disrupt it are limited by competition and immune regulation. The early microbiome also influences how the digestive system responds to food later in life. By participating in digestion from an early age, microbes help establish metabolic pathways that persist into adulthood. This does not lock the system into a single pattern, but it does provide a foundation upon which later adaptations are built. Throughout this early period, change occurs gradually. Sudden shifts are uncommon unless driven by major disturbances. This gradual pace allows the system to absorb variation without losing function. It also allows the body and microbiome to remain in dialogue, adjusting to one another as both develop. As sleep plays a central role in early growth, so too does rest support microbial development. During periods of rest, energy can be allocated toward maintenance and growth. Microbial activity continues quietly, processing nutrients and contributing to internal balance. Even in infancy, this unseen work supports the visible growth of the body. Looking back from adulthood, the beginnings of the microbiome are invisible yet enduring. The communities that form in early life do not remain unchanged, but they shape the system's capacity to adapt. They establish patterns of cooperation, resilience, and balance that persist even as specific members come and go. As you rest now, this early history remains part of you. The microbiome you carry is the result of countless small interactions, shaped slowly over time. It continues to adapt, just as it did in the beginning, responding gently to the conditions you provide. Nothing needs to be remembered. Nothing needs to be managed. The system knows how to continue. 7. Eating as a cooperative act. Eating is often experienced as a simple, personal act, guided by hunger and satisfaction. Yet beneath this familiar surface, it is also a deeply cooperative process. Every meal sets in motion a collaboration between human cells and microbial partners, each contributing according to their abilities. This cooperation does not require awareness or intention. It unfolds automatically, shaped by structure, chemistry, and time. When food enters the body, it brings with it a wide range of substances: proteins, fats, carbohydrates, fibers, minerals, and water. Human digestion begins the task of sorting and transforming these materials. Enzymes break large molecules into smaller ones. Muscles move food steadily along. Glands release secretions at precisely the right moments. These actions are efficient, but they are not complete on their own. Some components of food resist human digestion. Complex fibers and certain carbohydrates pass through the upper digestive tract largely unchanged. Rather than being wasted, these compounds become invitations for microbial participation. In the lower reaches of the gut, microbes take up the work where human enzymes leave off. This handoff is seamless, a quiet transition rather than a sharp boundary. From the microbial perspective, eating is an opportunity. Nutrients arrive regularly, carried by the movement of digestion. Microbes respond not to taste or preference, but to chemical availability. They metabolize what they can, extracting energy and releasing byproducts. These byproducts, in turn, influence the gut environment and support human cells. What begins as food for one becomes nourishment for another. This exchange illustrates the essence of cooperation. Neither human nor microbial digestion alone could achieve the same result. Together, they extract more value from food than either could independently. This shared efficiency has been favored by evolution, reinforcing the partnership across generations. The result is a system that conserves energy, reduces waste, and supports balance. The pace of eating influences this cooperation. Meals introduce nutrients in waves rather than all at once. As digestion proceeds, different compounds become available at different times. Microbes adjust their activity accordingly, responding to gradual changes rather than sudden abundance. This pacing supports stability, allowing microbial populations to remain balanced rather than fluctuating wildly. Chewing and swallowing begin this rhythm, breaking food into manageable portions. As digestion continues, movement slows in regions where microbial activity is highest. This slowing is not accidental. It allows time for fermentation and metabolic exchange. The body provides the setting. Microbes provide the processing. Together, they complete the transformation. The cooperative nature of eating extends beyond digestion itself. Signals generated during microbial metabolism influence appetite regulation and energy use. Chemical messengers produced in the gut interact with nerves and hormones, subtly shaping how the body responds to food. These signals do not dictate behavior, but they contribute to the overall regulation of intake and metabolism. Importantly, cooperation does not imply equality of control. The body maintains the environment, setting limits on growth and activity. Microbes respond within these boundaries, adapting to conditions provided. If balance shifts too far, regulatory mechanisms intervene, restoring equilibrium. This oversight ensures that cooperation remains beneficial rather than disruptive. Variety in food supports this cooperative system. Different foods introduce different substrates, encouraging diverse microbial activity. Diversity within the microbiome enhances resilience, allowing the system to handle variation without losing function. Over time, patterns of eating shape microbial communities, guiding them toward configurations that reflect available resources. This shaping occurs gradually. The microbiome does not change dramatically after a single meal. Instead, repeated patterns reinforce certain populations while allowing others to recede. This slow adaptation mirrors ecological processes in larger environments, where sustained conditions rather than brief events determine long-term structure. The act of eating also reflects cooperation across time. Their presence is the result of countless previous meals, habits, and adaptations. Each act of eating contributes incrementally to this ongoing relationship, reinforcing patterns established long before conscious awareness. During rest, the cooperative nature of eating becomes especially apparent. Food consumed earlier continues its journey, processed quietly by human and microbial systems alike. There is no pause in cooperation, only a shift in emphasis. Movement slows, but metabolism continues. Microbes remain active, completing tasks that require time rather than speed. This nighttime activity supports maintenance and repair. Byproducts of microbial digestion nourish the gut lining, supporting cellular renewal. Water absorption continues, contributing to fluid balance. The system works steadily, preparing the body for the next period of activity. Understanding eating as a cooperative act can soften the way we think about digestion. It highlights continuity rather than control, partnership rather than performance. The body is not extracting value from food alone. This perspective also emphasizes patience. Digestion is not meant to be rushed. When given time, the system functions smoothly. When rhythms are consistent, cooperation deepens. Stability emerges from repetition and balance. As you rest, this cooperation continues within you. Meals from earlier hours are still being processed, transformed through shared effort. Human cells and microbial partners remain engaged in their quiet exchange, sustaining balance without interruption. Nothing needs to be adjusted or directed. The process knows its course. Eating, then, is more than nourishment. It is participation in an ongoing biological partnership, one that has supported human life across generations. Each meal renews this partnership, reinforcing patterns of cooperation refined over time. As sleep deepens, this shared work continues, calm and reliable, carrying you gently through the night. 8. Breaking down what we cannot digest alone. Not everything we eat is meant to be handled by human digestion alone. Some parts of food are resistant by nature, built from complex structures that human enzymes cannot easily dismantle. These substances move quietly through the upper digestive tract, unchanged in form yet full of potential. Rather than being discarded, they are passed onward to a different set of partners, ones uniquely equipped for the task. Dietary fiber is one of the most familiar examples of this kind of material. Found in plants, fiber is made of complex carbohydrates arranged in patterns that human digestive enzymes cannot break apart. From the body's perspective, fiber is not useless. It is simply unfinished. Its transformation requires tools that human cells do not possess, but that certain microbes do. As fiber and other resistant compounds reach the large intestine, they enter an environment designed for patience. Movement slows, oxygen levels drop, and time becomes available. These conditions are ideal for microbial metabolism. Here, microbes encounter substances that have passed through the digestive system largely intact, carrying stored chemical energy waiting to be released. Microbes approach this task gradually. They attach enzymes to the surfaces of complex molecules, breaking them down piece by piece. This process is known as fermentation, though it bears little resemblance to anything dramatic or sudden. It unfolds slowly, over hours, guided by chemical gradients rather than force. Each step releases small fragments that can be used by microbes themselves or passed along to others. This breakdown is rarely the work of a single organism. Instead, it is a shared effort among many species. One microbe may begin the process, loosening a molecular structure. Another may continue, breaking the fragment into smaller units. Still, others may consume the byproducts. This layered cooperation allows the community to extract value from substances that would otherwise remain inaccessible. As microbes ferment fiber, they produce short-chain fatty acids and other small molecules. These compounds do not remain isolated within the gut. They are absorbed by the cells lining the colon and enter circulation, where they influence metabolism and support energy balance. In this way, microbial activity extends its reach beyond the digestive tract, contributing quietly to the body as a whole. The production of these byproducts also shapes the gut environment itself. Acidity levels shift slightly, discouraging the growth of harmful organisms while supporting beneficial ones. The intestinal lining receives nourishment, strengthening its integrity. The result is not only efficient digestion, but a stable internal setting that supports long-term balance. This process highlights an important truth about digestion. Efficiency does not come from speed. It comes from completeness. By allowing certain materials to move slowly and be processed by microbes, the body extracts more value from food than it otherwise could. Nothing is rushed. Nothing is wasted. Each component is handled according to its nature. The breakdown of resistant compounds also illustrates how digestion is distributed across space. Human enzymes dominate early stages, where conditions are suited to rapid chemical reactions. The digestive tract provides both environments, arranged in sequence, allowing each participant to work where it is most effective. This division of labor has been refined over evolutionary time. Organisms that could cooperate in this way gained an advantage, extracting more nourishment from available food. Over generations, this partnership became embedded in physiology, shaping both human digestion and microbial metabolism. What now seems ordinary is the result of countless adaptations layered upon one another. The presence of fiber and other resistant compounds also influences how the digestive system feels and functions. These materials add bulk and structure, affecting movement through the gut. They slow transit just enough to allow thorough processing without stagnation. This physical presence supports regularity and balance, contributing to the gentle rhythm of digestion. Importantly, microbial fermentation does not produce the same results from every substance. Different fibers are broken down in different ways, supporting different microbial populations. This variation encourages diversity within the microbiome, strengthening the system as a whole. Diversity ensures that no single pathway dominates, allowing flexibility and resilience. Over time, repeated exposure to certain compounds encourages the growth of microbes suited to processing them. The microbiome adapts gradually, learning from experience. This learning is not conscious, but biological. Populations shift subtly, reflecting the resources consistently available. In this way, long-term eating patterns shape microbial capabilities. Yet even as adaptation occurs, the system remains open to change. New compounds introduce new challenges, and the microbiome responds by adjusting composition and activity. This adaptability ensures that digestion remains functional across varied diets and environments. It is a system built for continuity rather than rigidity. During rest, the breakdown of these resistant compounds continues quietly. Food consumed earlier in the day may still be undergoing fermentation, releasing energy slowly and steadily. This prolonged processing supports sustained metabolic balance, avoiding sharp fluctuations. As you rest, microbes continue their work without interruption. They do not distinguish between day and night, only between availability and scarcity. When substrates are present, they metabolize. When they are gone, activity slows. This responsiveness keeps the system aligned with current conditions. Understanding this stage of digestion can bring a sense of appreciation for processes that operate beyond awareness. The body does not demand that everything be immediately useful. It allows certain things to wait, trusting that the right partners will complete the task. This patience is built into the structure of the digestive tract itself. Breaking down what we cannot digest alone is not a limitation, but a strength. It reflects a willingness to rely on cooperation, to share work across biological boundaries. It shows how human physiology is shaped not only by what it can do independently, but by what it can do together. As sleep deepens, this cooperation continues. Microbial enzymes remain active, molecules are transformed, and energy is released in measured amounts. The body receives what it needs without disturbance. Nothing calls for attention. The process flows onward, steady and calm. Within you, this quiet transformation unfolds, guided by time and partnership. It is one more example of how life sustains itself through cooperation rather than force. As you rest, this shared work carries on. 9. Fermentation and gentle energy production. Within the quiet depths of the large intestine, fermentation unfolds as a slow and steady process, one that rarely draws attention yet plays a meaningful role in sustaining balance. Unlike the rapid chemical reactions that occur earlier in digestion, fermentation is unhurried. It depends not on speed, but on time, patience, and cooperation among countless microscopic participants. Fermentation begins when microbes encounter substances that have resisted earlier stages of digestion. These compounds arrive intact, carrying complex chemical bonds that still hold energy. Microbes recognize these materials not as waste, but as opportunity. Through a series of carefully regulated steps, they begin to extract energy, breaking large molecules into smaller ones that can be used and shared. This process does not produce heat or motion that can be felt. It does not demand oxygen or dramatic change. Instead, it proceeds quietly, guided by enzymatic reactions that unfold at a pace suited to the environment. The absence of oxygen in the colon is not a limitation here, but a defining feature. It shapes which microbes participate and how they generate energy. As fermentation progresses, one of its primary outcomes is the production of short-chain fatty acids. These molecules are small, simple, and highly useful. They are absorbed by the cells lining the colon, where they provide a direct source of energy. In doing so, they support the maintenance and renewal of this vital tissue, helping preserve the integrity of the gut barrier. The influence of these fatty acids extends beyond the intestine. Once absorbed, they enter circulation and contribute to broader metabolic processes. They interact with liver function, influence how the body uses energy, and help regulate internal balance. Their effects are subtle, cumulative, rather than immediate, aligning with the slow nature of their production. Fermentation also produces gases as byproducts. These gases are part of the normal process and reflect microbial activity rather than dysfunction. The body manages them through absorption, transport, and eventual release, maintaining comfort and balance. This handling is efficient and largely unnoticed, another example of how the system accommodates its own processes. The microbial communities responsible for fermentation are diverse. Different species specialize in different substrates, and their metabolic pathways intersect. One organism's byproduct may become another's starting material. This interconnectedness ensures that energy extraction is thorough and that resources are used efficiently. This layered cooperation creates stability. When conditions shift slightly, such as changes in available substrates, microbial activity adjusts without abrupt disruption. The system absorbs variation smoothly, redistributing effort among participants. This adaptability is one reason fermentation remains reliable across a wide range of diets and routines. The gentle energy produced through fermentation complements energy obtained from other sources. Rather than causing sharp rises or falls, it provides a steady contribution. This steadiness supports metabolic equilibrium, especially during periods when food intake is paused. During sleep, for example, fermentation continues to supply small amounts of energy, supporting maintenance rather than activity. The timing of fermentation aligns naturally with rest. As external demands decrease, internal processes focus on consolidation and repair. Microbial activity persists, but without urgency. Energy is released gradually, matching the body's reduced requirements. This alignment contributes to the sense of continuity that defines healthy physiology. Fermentation also influences the physical environment of the gut. The acids produced help maintain an internal setting that favors beneficial microbes and discourages those less suited to coexistence. This chemical shaping reinforces balance without requiring constant immune intervention. The environment itself becomes a regulator. The cells lining the colon respond to fermentation products by adjusting their behavior. Gene expression shifts, mucus production adapts, and cellular turnover remains well regulated. These responses strengthen the gut's barrier function and support communication between the digestive system and the rest of the body. All of this occurs quietly, without conscious awareness. Over time, fermentation patterns reflect long-term habits. Regular exposure to certain fibers encourages the growth of microbes adept at processing them. This learning is gradual, encoded not in memory but in population dynamics. The microbiome becomes better suited to the diet it encounters most often, enhancing efficiency and comfort. Despite this specialization, the system remains flexible. Introducing new substrates leads to gradual shifts rather than immediate change. Microbes capable of handling the new material increase, while others recede. Balance is restored through adaptation rather than force. This flexibility allows digestion to remain stable even as life changes. Fermentation is often described as a secondary process, yet its contribution is foundational. It completes digestion where human enzymes cannot, ensuring that energy stored in complex compounds is not lost. It also supports internal environments that promote health and resilience. Without fermentation, digestion would be incomplete and balance harder to maintain. As you rest, fermentation continues without interruption. Microbes work steadily, transforming compounds that arrived hours earlier. Energy is released in small increments, supporting the body's quiet needs. The process does not wake you or demand attention. It simply continues, guided by availability and time. This ongoing activity illustrates a broader principle of biology, that not all productivity is visible or immediate. Some of the most important work happens slowly, in the background, sustained by cooperation and patience. Fermentation exemplifies this principle, turning what seems inert into something useful through time alone. Within you, this gentle energy production supports continuity. It bridges periods of eating and fasting, activity and rest. It smooths transitions, preventing abrupt changes. In doing so, it contributes to the body's remarkable ability to maintain equilibrium across the day and night. As sleep deepens, fermentation remains part of the quiet hum of life within. Molecules are transformed, energy is shared, and balance is preserved. Nothing needs to be managed or monitored. The system knows its role, performing it steadily and reliably. 10 microbes and nutrient availability. Nutrients are often described as things the body takes in, absorbs, and uses. Yet this description captures only part of the story. Between the moment food enters the digestive tract and the moment nutrients reach the cells that need them, there is a long and quiet process of preparation. Within this process, the microbiome plays a subtle but important role. As food moves through the digestive system, human enzymes break large molecules into smaller ones, preparing them for absorption. Proteins become amino acids, fats are separated into fatty acids, and simple carbohydrates are reduced to sugars. These transformations are precise and efficient, yet they do not address every component of food. Many nutrients exist in forms that require additional steps before they can be fully utilized. This is where microbial activity becomes especially significant. Microbes interact with nutrients that remain bound within complex structures, releasing them slowly over time. In doing so, they help expand the range of substances the body can access. Rather than acting as competitors for nutrients, these microbes function as facilitators, unlocking potential that would otherwise remain inaccessible. One clear example of this facilitation is the microbial production of certain vitamins. Some vitamins are present in food, while others are synthesized within the gut itself. Microbial communities are capable of producing compounds such as vitamin K and several B vitamins. These substances are absorbed locally and contribute to normal physiological processes, quietly supplementing what comes from the diet. This production is not excessive or uncontrolled. It occurs at a pace aligned with microbial metabolism and environmental conditions. The body absorbs what it needs, and balance is maintained through regulation and feedback. Nothing about this exchange is abrupt. It unfolds gradually, matching the overall rhythm of digestion. Microbes also influence how minerals are made available. Certain dietary components bind minerals, limiting their absorption. Microbial activity can modify these compounds, reducing their binding capacity and allowing minerals to be absorbed more efficiently. This indirect contribution helps ensure that essential elements remain accessible without overwhelming the system. The timing of nutrient release is as important as the quantity. Rapid absorption can lead to sharp fluctuations, while slow, steady availability supports equilibrium. Microbial processing tends to favor gradual release, smoothing transitions and supporting stable metabolic conditions. This steadiness aligns well with the body's preference for balance over extremes. The cells lining the intestine respond to microbial byproducts by adjusting transport mechanisms. When certain compounds are present, absorption pathways become more active. When they are scarce, activity slows. This responsiveness ensures that nutrient uptake remains proportional to availability, avoiding unnecessary strain. The microbiome also influences the environment in which absorption occurs. By shaping acidity and producing supportive compounds, microbes help maintain conditions that favor efficient transport across the intestinal lining. These environmental adjustments are subtle, yet they have lasting effects on how nutrients move from the gut into circulation. Over time, repeated patterns of eating shape this process. The microbiome adapts to the nutrients it encounters most often, becoming more efficient at processing familiar compounds. This adaptation does not lock the system into rigidity, but it does enhance reliability. The body becomes better at extracting nourishment from its usual sources. Despite this specialization, flexibility remains. When new foods are introduced, microbial populations shift gradually, exploring new metabolic pathways. Nutrient availability adjusts accordingly, allowing the system to expand its capabilities. This adaptability is one reason humans can thrive on diverse diets across cultures and environments. Nutrient availability also influences microbial behavior in return. When certain nutrients are abundant, microbial growth patterns adjust. When they are scarce, activity slows. This mutual responsiveness creates a feedback loop, aligning microbial metabolism with the body's current state. The result is coordination rather than competition. During periods of rest, this coordination becomes especially apparent. Nutrients absorbed earlier continue to circulate, supporting maintenance and repair. Microbial processing continues quietly, releasing compounds at a pace suited to rest rather than activity. This gentle supply supports cellular renewal without demanding attention or effort. Sleep provides an ideal context for this steady exchange. The digestive tract focuses on absorption and maintenance, while microbes continue their work in the background. Nutrients are distributed where needed, supporting balance across systems. This ongoing contribution highlights an important aspect of the microbiome's role. It does not act as a reserve or emergency system, but as a constant presence, shaping nutrient flow moment by moment. Its influence is cumulative rather than dramatic, building stability through consistency. Understanding nutrient availability in this way can soften expectations around digestion and metabolism. The body does not extract everything immediately, nor does it require constant input. Instead, it relies on layered processes that extend nourishment over time. Microbial activity is central to this extension, ensuring continuity between meals. This continuity supports resilience. By smoothing transitions and distributing nutrients gradually, the system reduces stress on metabolic pathways. Energy levels remain steadier, and internal balance is easier to maintain. The body benefits not from abundance alone, but from measured availability. As you rest, nutrients absorbed earlier are still being used. Microbial contributions continue to shape what is available and when. There is no clear boundary between digestion and metabolism, only a gentle flow from one stage to the next. Everything proceeds according to rhythm rather than command. Within you, this quiet exchange sustains life in small, steady increments. Cells receive what they need, microbes respond to what is present, and balance is preserved. Nothing needs to be accelerated or optimized. The system functions as it has evolved to do, guided by cooperation and time. As sleep deepens, nutrient availability remains aligned with rest. Energy is supplied slowly, supporting repair rather than exertion. The microbiome continues its subtle role, neither visible nor silent, but quietly present. Through this shared work, the body maintains its equilibrium, carrying you gently through the night. 11. The intestinal lining as a living boundary. Within the long passage of the digestive tract, there exists a boundary that is both delicate and resilient, both selective and welcoming. This boundary is the intestinal lining, a living interface between the external world of food and microbes and the internal world of the human body. Though thin and unseen, it performs one of the most important balancing acts in human physiology, quietly deciding what may pass through and what must remain outside. The intestinal lining is not a static wall. It is composed of living cells arranged in a continuous layer, renewed constantly over time. These cells are born, mature, perform their functions, and are replaced in a steady cycle. This ongoing renewal ensures that the boundary remains intact despite constant exposure to mechanical movement, chemical digestion, and microbial activity. It is a surface designed not for permanence, but for adaptability. As food is broken down into absorbable components, these nutrients approach the intestinal lining from the gut's interior. The lining responds selectively, allowing small, useful molecules such as sugars, amino acids, fatty acids, vitamins, and minerals to pass through. Larger structures, including most microbes, are kept at a careful distance. This selectivity is not rigid, but finely tuned, guided by molecular recognition rather than force. Between the intestinal cells lie tight junctions, structures that regulate the spaces through which substances may pass. These junctions open and close subtly, responding to chemical signals and physiological needs. When conditions are favorable, absorption is efficient. When conditions change, permeability adjusts. This dynamic control allows the body to remain responsive without becoming vulnerable. Covering the intestinal lining is a layer of mucus, secreted continuously by specialized cells. This mucus is not merely protective, it is also a habitat. Certain microbes reside within it, forming a buffer zone between the gut contents and the epithelial cells beneath. This arrangement allows microbes to remain close enough to interact and contribute, while maintaining a respectful separation that preserves tissue integrity. The mucus layer varies along the digestive tract, becoming thicker in regions where microbial density is highest. In the colon, where fermentation occurs and microbial populations are dense, this layer provides both nourishment and protection. Microbes adapted to this environment interact with mucus components, while the underlying cells remain shielded from direct contact. This spatial organization supports coexistence rather than conflict. The intestinal lining is also an active communicator. Cells here sense chemical signals produced by microbes and respond accordingly. They adjust mucus production, alter absorption rates, and release signaling molecules that influence immune activity. These responses are measured and proportional, ensuring that balance is maintained without excessive reaction. Embedded within and beneath the intestinal lining is a large portion of the immune system. This placement reflects the importance of vigilance at this boundary. Immune cells sample microbial signals, learning to distinguish between familiar partners and potential threats. Most of the time, this surveillance results in tolerance rather than action. The goal is not elimination, but peaceful coexistence. When microbes behave within expected patterns, immune responses remain calm. When signals indicate imbalance, regulatory mechanisms activate to restore order. This may involve subtle shifts in microbial populations rather than overt inflammation. Such restraint preserves the integrity of the boundary and avoids unnecessary disruption. The intestinal lining also plays a role in educating the immune system over time. Early exposure to microbial signals helps shape immune responses, promoting tolerance and reducing overreaction. This education continues throughout life as the microbiome and immune system remain in dialogue. The lining serves as the classroom for this ongoing lesson. Nutrient absorption and immune regulation are not separate tasks here. They are integrated functions of the same living surface. As nutrients pass through the lining, immune cells monitor accompanying signals. As microbes produce byproducts, epithelial cells interpret their meaning. This integration allows the body to respond appropriately to what it encounters. The health of the intestinal lining depends on support from both human and microbial sources. Microbial fermentation produces compounds that nourish epithelial cells, strengthening their function. In return, the lining maintains conditions that support beneficial microbes. This mutual reinforcement stabilizes the boundary, making it more resilient to change. Stress on the system is inevitable, yet the lining is designed to recover. Its rapid turnover allows damaged cells to be replaced quickly. Its regulatory mechanisms allow permeability to adjust as needed. These features ensure that temporary disturbances do not lead to lasting imbalance. Recovery is built into the structure. During rest, particularly during sleep, the intestinal lining continues its quiet work. Cellular renewal proceeds, supported by steady nutrient availability. Mucus secretion continues, maintaining the protective layer. Immune surveillance remains alert but calm. This nighttime maintenance supports the boundary's integrity for the day ahead. The lining does not distinguish between conscious and unconscious states. It responds only to internal conditions, adjusting continuously. In sleep, when external inputs pause, internal processes take precedence. Repair and renewal occur without interruption, guided by biological rhythms rather than awareness. Seeing the intestinal lining as a living boundary rather than a passive barrier changes how digestion is understood. It is not merely a point of transfer, but a site of decision-making and communication. It mediates relationships between the body and its microbial partners, ensuring cooperation remains beneficial. This boundary is neither fragile nor impenetrable. It is flexible, responsive, and self-renewing. Its strength lies in its ability to adapt rather than resist, to regulate rather than block. Through this adaptability, it maintains balance across changing conditions. As you rest, this living boundary continues to function with quiet precision. Nutrients pass through as needed. Microbes remain at an appropriate distance. Signals are exchanged, interpreted, and acted upon. Nothing is hurried. Nothing is neglected. The system performs its role, as it always has. Within you, this boundary supports continuity, allowing interaction without intrusion. It is one of the many unseen structures that make life possible, working without recognition or command. As sleep deepens, it remains steady, preserving balance and harmony through the night. 12. Microbial support of the immune system. Within the quiet environment of the gut, the immune system carries out much of its work, not through confrontation, but through careful observation and restraint. A significant portion of the body's immune cells reside in and around the digestive tract, positioned where the external world meets the internal environment. Here, microbes and immune cells exist in close proximity, engaged in a continuous, subtle exchange that shapes immune behavior over a lifetime. The immune system is often described as a defense force designed to identify and eliminate threats. While this is one of its functions, it is only part of the story. Equally important is the ability to tolerate what is familiar and beneficial. The microbiome plays a central role in teaching this distinction, helping the immune system learn when to remain calm and when to respond. From early life onward, immune cells are exposed to microbial signals produced by the gut microbiome. These signals are not alarms, but information. They indicate presence, activity, and balance. Immune cells interpret this information and adjust their responses accordingly. Over time, this repeated exposure builds a pattern of recognition, allowing the immune system to respond with precision rather than force. The gut provides an ideal setting for this education. Microbes are abundant, diverse, and relatively stable. They produce a wide range of molecular signals, reflecting different metabolic activities. Immune cells sample these signals continuously. This familiarity reduces unnecessary immune activation and supports long-term balance. Rather than being suppressed, the immune system is guided. Regulatory immune cells play a key role in this process, moderating responses and promoting tolerance. These cells help ensure that immune activity remains proportional, preventing excessive inflammation that could damage tissues or disrupt microbial communities. The result is a state of readiness without constant aggression. Microbial byproducts contribute directly to this regulatory environment. Compounds produced during fermentation and other metabolic processes influence immune cell behavior, encouraging calm responses and supporting tissue health. These chemical interactions are gentle and ongoing, reinforcing a cooperative relationship rather than triggering defense mechanisms. The immune system also influences the microbiome in return. By shaping the gut environment through signaling and regulation, immune cells help maintain microbial balance. They discourage the overgrowth of organisms that could disrupt stability while supporting the persistence of beneficial populations. This influence is subtle, operating through environmental conditions rather than direct elimination. This mutual shaping creates a feedback loop. Microbes influence immune development, and immune activity shapes microbial communities. Neither system operates independently. Instead, they adapt together, forming a partnership that supports resilience and flexibility. This partnership allows the body to coexist with a vast microbial population without constant conflict. The intestinal lining serves as the meeting place for this interaction. Immune cells positioned just beneath the epithelial surface sample microbial signals that pass through or are presented by epithelial cells. This indirect communication preserves separation while allowing information exchange. The boundary remains intact, yet communication continues. Over time, this dialogue influences how the immune system responds beyond the gut. Signals originating in the digestive tract can affect immune behavior elsewhere in the body, shaping systemic responses. In this way, the microbiome contributes to immune regulation at a broader scale, extending its influence beyond its immediate environment. Importantly, this system is not static. Immune responses adapt as microbial populations change. Dietary shifts, environmental exposure, and aging all influence the microbiome, and the immune system adjusts accordingly. This adaptability prevents rigid responses that could lead to imbalance. Flexibility becomes a form of strength. Periods of rest play an important role in maintaining this balance. During sleep, immune activity shifts toward maintenance and regulation rather than rapid response. Microbial signals continue to be processed, reinforcing patterns of tolerance and calm. This nighttime phase supports immune learning and stabilization. The immune system's relationship with the microbiome also reflects a broader principle of biology, that exposure, when gradual and controlled, builds resilience. Rather than isolating the body from all external influences, the immune system learns through interaction. The microbiome provides a constant, manageable source of information, allowing immune cells to refine their responses over time. This refinement reduces the likelihood of overreaction. When the immune system is accustomed to a diverse range of microbial signals, it becomes better at distinguishing genuine threats from harmless presence. This discrimination protects tissues from unnecessary inflammation and preserves energy for situations that truly require response. The presence of the microbiome also supports immune memory. Repeated interactions reinforce regulatory pathways, making balanced responses more likely in the future. This memory is not conscious or deliberate. It is encoded in cellular behavior and signaling patterns, shaped gradually through experience. As you rest, this immune-microbial partnership continues its quiet work. Immune cells monitor signals, interpret patterns, and adjust behavior. Microbes continue their metabolic activity, producing the signals that guide this process. There is no urgency, no alarm, only steady communication. Sleep provides a context in which immune regulation can proceed without distraction. With fewer external challenges, the immune system focuses on calibration rather than defense. The microbiome remains an active participant, reinforcing the conditions that support tolerance and balance. Understanding microbial support of the immune system highlights how health is maintained through cooperation rather than isolation. The body does not aim to exclude all microbes, nor does it surrender control. Instead, it establishes a relationship built on communication and regulation. This relationship is maintained quietly through countless small interactions that accumulate over time. Within you, this partnership has been shaped since early life, refined through daily experience. It continues now, even as you rest. Immune cells and microbes remain in dialogue, sustaining balance without effort or awareness. Nothing needs to be managed or directed. As sleep deepens, this quiet exchange persists, supporting resilience and continuity. The immune system remains calm, informed by familiar signals. The microbiome remains active, contributing to regulation rather than disruption. Together, they maintain harmony within the body, carrying you gently through the night. 13. The gut and the nervous system. Deep within the body, far from conscious thought, the digestive system and the nervous system remain in constant communication. This exchange is quiet, steady, and continuous, forming a bridge between the activity of the gut and the broader regulation of the body. It is not a conversation of words or awareness, but one of signals, rhythms, and chemical messages that travel along well-established pathways. The digestive tract is richly supplied with nerves, forming an extensive network embedded within its walls. This network is often referred to as the enteric nervous system, a collection of neurons capable of sensing, responding, and coordinating activity locally. It monitors movement, pressure, and chemical composition, adjusting digestion moment by moment. Much of this activity occurs independently of conscious control, allowing digestion to proceed whether attention is present or not. Although the enteric nervous system can function on its own, it is not isolated. It remains closely connected to the central nervous system through long nerve pathways, most notably the vagus nerve. This connection allows information to travel between the gut and the brain, integrating digestion with overall physiological state. Signals move in both directions, creating a loop rather than a one-way command structure. From the gut to the brain, sensory information flows continuously. Stretch receptors respond to movement and fullness. Chemical receptors detect the presence of nutrients and microbial byproducts. These signals inform the nervous system about the state of digestion, allowing broader systems to adjust energy use, circulation, and rest. The brain does not need to interpret each signal consciously. Instead, it responds through automatic regulation. Microbial activity contributes significantly to this signaling environment. As microbes ferment compounds and metabolize nutrients, they produce substances that interact with nerve endings in the gut. Some of these compounds influence the release of neurotransmitters and neuromodulators, shaping the messages sent along neural pathways. This influence is subtle, cumulative, and constant. The nervous system, in turn, influences the gut environment. Through neural signaling, it adjusts muscle contractions, secretion rates, and blood flow. These adjustments shape the conditions under which microbes operate, influencing nutrient availability and movement. This reciprocal influence ensures that neither system acts alone. Each responds to the state of the other. One of the most remarkable features of this connection is its steadiness. Signals do not spike abruptly under normal conditions. Instead, they reflect gradual changes, allowing the system to adapt smoothly. This steadiness supports balance, preventing unnecessary strain on either digestion or neural regulation. The gut-nervous system connection also plays a role in timing. Digestive activity follows rhythms aligned with daily patterns of eating and rest. Neural signals help coordinate these rhythms, slowing digestion during sleep and increasing readiness during waking hours. Microbial activity adjusts accordingly, responding to changes in movement and substrate availability. During sleep, the nature of communication shifts, external sensory input decreases, and internal signals become more prominent. The nervous system reduces its focus on rapid response and directs more attention toward maintenance. Digestive signaling continues, but with an emphasis on regulation rather than reaction. This environment supports calm, restorative processes. Microbes continue to participate during this phase. Their metabolic activity generates signals that reinforce steady neural regulation. These signals do not provoke alertness or arousal. Instead, they contribute to a background state that supports rest. The system remains active without becoming stimulating. The gut-brain connection is often discussed in dramatic terms, yet its everyday function is quiet and consistent. It does not exist to produce strong sensations or conscious experiences. Its primary role is coordination, ensuring that digestion, metabolism, and neural regulation remain aligned. Most of the time, this coordination goes unnoticed, precisely because it works well. The presence of the microbiome adds depth to this connection. Microbes expand the range of signals available, enriching the information the nervous system receives. This enrichment allows for finer adjustments, supporting stability across varied conditions. It also means that digestion is not merely mechanical, but communicative. Over time, repeated patterns strengthen these connections. Regular rhythms of eating and resting reinforce predictable signaling. The nervous system learns what to expect, and the microbiome adapts to provide consistent input. This familiarity reduces the need for strong regulatory responses, allowing the system to operate smoothly. Disruption is possible, but the system is designed to recover. Gradual changes are absorbed through adaptation. Sudden shifts may require temporary adjustment, but balance is restored through feedback rather than force. The connection between gut and nervous system supports this resilience by maintaining open communication. As you rest now, this communication continues uninterrupted. Signals travel quietly along nerves. Microbial byproducts influence neural activity without drawing attention. The nervous system responds by maintaining steady rhythms. Nothing demands awareness. Nothing requires intervention. Sleep offers an ideal setting for this coordination. With fewer competing signals from the external world, internal communication becomes more efficient. The gut continues its work, and the nervous system supports it by maintaining calm, stable conditions. This partnership contributes to the restorative quality of rest. The relationship between the gut and nervous system reflects a broader principle of biology, that regulation often depends on dialogue rather than control. Instead of issuing commands, systems exchange information and adjust accordingly. This approach conserves energy, reduces strain, and supports long-term stability. Within you, this dialogue has been ongoing since early development. It has adapted to your routines, your environment, and your patterns of rest. It continues now, even as awareness fades. The signals remain steady, the response is measured. As sleep deepens, digestion and neural regulation move in harmony. The microbiome remains an active participant, contributing signals that support balance. The nervous system remains attentive without becoming alert. Nothing needs to be directed. Nothing needs to be solved. The system operates as it was shaped to do, guided by rhythm and communication. Within the quiet of sleep, this connection sustains you, moment by moment, through the night. 14 daily rhythms of digestion and microbial activity. Every living system is shaped by rhythm, and within the human body, digestion follows a steady cadence that unfolds across the day and night. These rhythms are not imposed from outside, but emerge naturally from repeated patterns of eating, resting, and activity. Within this temporal structure, the microbiome adapts its behavior, aligning microbial activity with the body's internal clock in a quiet and reliable way. Digestion does not proceed at a constant pace. Instead, it rises and falls in response to signals that reflect time of day, availability of food, and state of rest. When meals are consumed, digestive activity increases. Muscular movement becomes more pronounced, secretions are released, and microbial metabolism accelerates as new substrates arrive. When eating pauses, activity gradually shifts toward processing and maintenance. These daily patterns are coordinated by circadian rhythms, internal cycles that regulate physiology over approximately 24 hours. Cells throughout the body respond to these cycles, including those of the digestive tract. Enzyme production, intestinal movement, and blood flow fluctuate predictably, creating windows of heightened and reduced activity. This coordination ensures that digestion remains efficient without becoming overwhelming. Microbes living within the gut are sensitive to these rhythms as well. They do not perceive time as humans do, yet they respond to environmental cues shaped by circadian regulation. Changes in nutrient availability, acidity, and movement signal when to increase or reduce activity. Over time, microbial populations synchronize their metabolic patterns to the host's daily routine. This synchronization benefits both sides. For the body, aligned microbial activity supports efficient digestion and nutrient absorption during active periods, while promoting gentle processing during rest. For microbes, predictable rhythms provide stability, allowing populations to allocate resources efficiently rather than responding to constant unpredictability. Eating patterns play a central role in establishing these rhythms. Regular meal timing creates recurring waves of nutrients that travel through the digestive tract. Microbes respond by organizing their activity around these waves, increasing metabolism when substrates are present and conserving energy when they are not. This responsiveness reduces waste and supports balance. During waking hours, digestion is often more active. Movement, posture, and neural input support the forward motion of food. Microbial fermentation begins later in the digestive process, extending beyond the immediate post-meal period. This staggered timing spreads metabolic activity across hours rather than concentrating it all at once. As evening approaches and external activity decreases, digestion begins to slow. Neural signaling shifts toward rest, and muscular contractions become gentler. Yet, digestion does not stop. Food consumed earlier continues its journey, and microbial fermentation remains active. This continuation ensures that nutrients are extracted thoroughly, even as the body prepares for sleep. Nighttime represents a distinct phase in digestive rhythm. With eating paused and movement reduced, the digestive tract enters a maintenance-oriented state. Absorption continues, fermentation proceeds, and cellular renewal accelerates. Microbes remain metabolically active, but their activity is shaped by the absence of new substrates and the body's reduced demands. This nocturnal phase highlights the importance of rhythm over intensity. Rather than processing everything immediately, the body allows digestion to unfold over extended periods. Microbial activity during the night contributes to steady energy availability, supporting repair and regulation rather than exertion. The cells lining the gut respond to these rhythms by adjusting their behavior. During rest, they focus on repair, strengthening the barrier, and renewing tissue. Microbial byproducts produced during fermentation support this renewal, creating a feedback loop that aligns microbial activity with host needs. Immune regulation also follows daily rhythms. During sleep, Immune responses shift toward tolerance and maintenance rather than rapid defense. Microbial signals reinforce this calm state, supporting balance at the gut boundary. This alignment reduces unnecessary activation and preserves energy. Over time, consistent daily rhythms reinforce stability within the microbiome. Populations learn when to expect nutrients and when to conserve resources. This learning is encoded not in memory, but in population dynamics and metabolic pathways. The result is a system that functions smoothly with minimal disruption. When rhythms are disrupted, adaptation occurs. Changes in meal timing or sleep patterns alter environmental cues, and microbes respond gradually. Some populations increase activity at new times, while others decline. Balance is restored through adjustment rather than force, reflecting the system's inherent flexibility. Despite this adaptability, regularity remains beneficial. Predictable rhythms reduce stress on digestive and microbial systems, allowing them to operate efficiently. The body does not require strict schedules, but it benefits from consistency. Repetition reinforces coordination, making digestion feel effortless. Rest plays a vital role in maintaining these rhythms. Sleep provides a protected interval during which maintenance can occur without competition from external demands. Microbial activity during sleep supports this maintenance, contributing to steady internal conditions. As you rest now, your digestive system is following these familiar patterns. Activity has slowed but not ceased. Microbes continue to ferment remaining substrates, releasing energy gradually. Cells absorb nutrients and renew themselves. These daily cycles have been shaped over evolutionary time, aligning human physiology with natural patterns of light and dark, activity and rest. The microbiome has adapted alongside, learning to operate within these constraints. Understanding digestion as a rhythmic process can bring a sense of reassurance. Nothing needs to be rushed or forced. The body knows when to act and when to rest. Microbial partners respond accordingly, supporting balance across the cycle. Within you, this rhythm continues, steady and reliable. Each phase flows into the next, guided by internal clocks rather than conscious direction. As sleep deepens, digestion and microbial activity remain aligned with rest, sustaining balance quietly through the night. 15. Diet as an ecological influence. Food does more than nourish the human body directly. Each meal also shapes the internal environment in which microbial life exists, subtly influencing which organisms thrive, which recede, and how the entire system functions over time. In this way, diet acts not as a command, but as an ecological influence, altering conditions and allowing the microbiome to respond according to its nature. An ecosystem does not change because one organism issues instructions to another. It changes because resources shift, environments adjust, and opportunities arise. The gut microbiome responds to diet in the same manner. Foods introduce particular compounds into the digestive tract, and microbes respond by adjusting their activity and growth. Those best suited to the available resources increase gradually, while others diminish without conflict. This process unfolds slowly. A single meal rarely produces lasting change. Instead, patterns of eating accumulate over days, weeks, and months, shaping microbial communities through repetition. The microbiome learns from experience, adapting to what is consistently present rather than reacting dramatically to brief variation. This gradual shaping supports stability and resilience. Different foods influence the gut environment in distinct ways. Some provide complex carbohydrates that require microbial fermentation. Others are more easily digested by human enzymes, leaving less work for microbes downstream. These differences affect how much substrate reaches the large intestine, where microbial activity is most concentrated. Over time, this influences which populations are most active. Fiber-rich foods, for example, tend to support a broader range of microbial activity. Because fiber resists human digestion, it arrives in the colon intact, offering nourishment to diverse microbial species. This diversity encourages a balanced ecosystem, where many organisms participate in processing and regulation. The result is not increased activity alone, but distributed activity that supports stability. Foods lower in resistant compounds may reduce the amount of substrate available for fermentation. Microbial activity adjusts accordingly, focusing on available resources. This adjustment is not harmful in itself, but it illustrates how diet shapes microbial roles rather than dictating outcomes. The system remains flexible, responding to what is provided. Importantly, the microbiome does not evaluate foods as good or bad. It responds only to chemical composition and availability. The ecological influence of diet emerges from this neutrality, as populations align with what they can use most effectively. The physical properties of food also matter. Texture, structure, and water content influence how food moves through the digestive tract. Slower movement allows more time for microbial processing, while faster movement limits it. These physical effects shape microbial opportunities as much as chemical composition does. Over time, consistent dietary patterns create predictable environments. Microbes adapt to these environments by refining metabolic pathways, increasing efficiency, and coordinating activity. This adaptation does not reduce flexibility, but it does enhance reliability. The system becomes well suited to familiar conditions. When dietary patterns change gradually, the microbiome adjusts smoothly. New substrates encourage the growth of organisms capable of processing them. This adaptation mirrors ecological succession in larger environments, where gradual changes allow communities to reorganize without collapse. Sudden changes may require more noticeable adjustment, yet the system is designed to recover. Diversity within the microbiome supports resilience, ensuring that essential functions continue even as populations reorganize. Diet also influences the chemical environment of the gut. Microbial metabolism produces compounds that affect acidity, which in turn shapes microbial survival. This feedback loop reinforces patterns established by diet, making certain conditions more stable over time. The result is an ecosystem shaped by interaction rather than control. The relationship between diet and the microbiome extends beyond digestion. Microbial byproducts influence immune regulation, gut barrier function, and metabolic signaling. These effects are indirect and cumulative, emerging from sustained patterns rather than immediate responses. Diet thus influences the body not only through nutrients, but through the ecological shifts it supports. During rest, the effects of diet continue to unfold. Food consumed earlier in the day remains in the digestive tract, undergoing microbial processing. Fermentation continues, releasing compounds that support maintenance and repair. The body benefits from this extended influence, which spreads the impact of meals across time. Sleep provides a context in which these processes proceed without interruption. With external demands reduced, internal systems can integrate the ecological effects of diet more fully. Microbial activity aligns with rest, contributing to steady internal conditions rather than rapid change. Understanding diet as an ecological influence encourages patience. The body does not respond instantly to every choice, nor does the microbiome transform overnight. Instead, change accumulates slowly, shaped by consistency rather than intensity. This perspective aligns with the body's preference for gradual adaptation. It also highlights cooperation rather than control. The microbiome is not directed, but influenced. The body provides an environment shaped by diet, and microbes respond according to their nature. This partnership supports balance through responsiveness rather than rigidity. As you rest now, the ecological influence of your recent meals continues quietly within you. Microbes metabolize available compounds, populations adjust subtly, and the gut environment reflects these interactions. Nothing needs to be managed or evaluated. Within you, diet has left its imprint not as a command, but as a condition. Microbial communities reflect the resources they encounter, adapting gradually to support digestion and balance. This adaptation continues over time, shaped by repetition and rhythm. As sleep deepens, these ecological processes proceed steadily. Fermentation continues, nutrients are released, and internal conditions remain stable. The influence of diet extends beyond waking hours, integrated into the body's ongoing maintenance. In this way, food becomes more than nourishment. It becomes part of an ongoing conversation between human cells and microbial partners, shaping an ecosystem through quiet influence rather than force. As rest continues, this conversation remains gentle and uninterrupted. 16. Change, diversity, and stability in the microbiome. Within the living system of the human gut, change is not an exception but a constant presence. Microbial populations shift from day to day, responding to subtle variations in diet, routine, and internal conditions. Yet despite this continual movement, the microbiome tends towards stability. This balance between change and continuity is one of its most defining features, allowing it to support digestion and health across a wide range of circumstances. Stability in biology does not mean stillness. It refers instead to the ability of a system to maintain function while accommodating variation. The microbiome exemplifies this principle. Individual species may increase or decrease in number, but essential roles such as fermentation, nutrient processing, and immune interaction continue uninterrupted. This functional stability emerges from diversity, where multiple organisms can perform similar tasks in slightly different ways. Diversity within the microbiome acts as a form of biological insurance. If one population declines, others can compensate. This redundancy ensures that digestion remains effective even as conditions change. It also allows the microbiome to recover from disturbances gradually, re-establishing balance without requiring precise restoration of its previous state. Change enters the system through many pathways. Dietary variation introduces new substrates. Daily rhythms alter timing and movement. Environmental exposure adds new microbial signals. Even internal factors such as hormonal fluctuations influence gut conditions. Each of these influences nudges the microbiome in small ways, encouraging adaptation rather than upheaval. The response to change is typically measured rather than immediate. Microbial populations adjust their growth rates gradually, reflecting sustained conditions rather than momentary events. This delay prevents overreaction and supports continuity. Short-lived changes often leave little lasting trace, while repeated patterns shape the ecosystem more deeply. One of the strengths of the microbiome lies in its capacity for self-organization. Microbes interact not only with the host but with one another, forming networks of cooperation and competition that regulate population balance. These interactions create internal checks and balances, preventing any single species from dominating indefinitely. The result is a dynamic equilibrium maintained through interaction rather than control. Environmental conditions within the gut further support this balance. Microbes adapted to these conditions thrive, while others remain limited. This environmental shaping reduces the likelihood of extreme shifts, reinforcing stability through physical and chemical means. Resilience is another key feature of this system. When disruption occurs, whether through dietary change, temporary stress, or environmental exposure, the microbiome tends to move toward a new stable state rather than collapsing. This new state may differ from the previous one, yet it often preserves core functions. Resilience allows the system to continue supporting digestion and regulation even as it adapts. Importantly, recovery does not require a return to an exact previous composition. Stability emerges from function, not from precise membership. As long as essential processes continue, the system can tolerate variation. This flexibility allows humans to adapt to diverse diets and environments without losing digestive capacity. Time plays a crucial role in maintaining this balance. Gradual change allows adaptation, while sudden extremes are buffered by diversity and redundancy. The microbiome's response unfolds over days and weeks rather than minutes, aligning with the body's preference for steady adjustment. This temporal buffering protects against volatility. The host body contributes to stability by maintaining consistent internal conditions. Temperature, hydration, and immune regulation provide a stable backdrop against which microbial variation occurs. This consistency allows microbes to focus on metabolic activity rather than survival, supporting efficient cooperation. Microbial diversity also influences how the immune system behaves. A diverse microbiome presents a wide range of familiar signals, helping the immune system calibrate its responses. This exposure supports tolerance and reduces the likelihood of excessive reaction. In this way, diversity contributes not only to digestive stability, but to immune balance as well. During rest, especially during sleep, the microbiome's stabilizing mechanisms continue to operate. Without the introduction of new food, activity shifts toward processing and maintenance. Populations adjust quietly, guided by internal interactions rather than external input. The body does not monitor these changes consciously, nor does it need to. Feedback mechanisms embedded in physiology and microbial interaction guide adjustment automatically. Signals are exchanged, conditions are sensed, and responses unfold without awareness. Understanding the microbiome in terms of change and stability can bring reassurance. Variation does not imply fragility. Instead, it reflects a capacity to respond. Stability does not require sameness, only continuity of function. The microbiome embodies this balance, maintaining support through adaptation rather than resistance. As you rest now, this balance continues within you. Microbial populations shift subtly, responding to the conditions provided. Diversity supports resilience, and resilience preserves stability. Nothing needs to be corrected or controlled. The system adjusts as it always has, guided by interaction and time. Within this quiet adaptability lies strength. The microbiome does not depend on perfection, only on cooperation. Its ability to change without losing function allows it to support the body through varied experiences. As sleep deepens, this adaptive stability remains in place, sustaining digestion and balance through the night. 17. From the earliest moments of life to the quiet years of old age, the microbiome remains a constant companion, changing not abruptly, but gradually, in step with the body itself. It does not follow a fixed path, nor does it reach a final, perfected state. Instead, it evolves gently across the lifespan, adapting to shifting physiology, habits, and environments, while preserving its essential role in balance and digestion. In early life, the microbiome is flexible and exploratory. Populations rise and fall quickly, responding to new exposures and nutritional inputs. This early variability reflects openness rather than instability. The developing body provides an environment still learning its own rhythms, and microbial communities adjust continuously, forming the foundations of long-term cooperation. Diversity increases steadily during childhood as exposure broadens and digestive capacity matures. As growth continues, the digestive system becomes more predictable. Patterns of eating, sleeping, and activity settle into routines. The microbiome responds by stabilizing its composition and function. While populations continue to shift, changes occur more slowly. Core functions become reliably established, supported by overlapping microbial roles. This period marks the emergence of a mature microbial ecosystem. During adolescence and early adulthood, internal conditions change once again. Growth slows, metabolism adjusts, and hormonal patterns shift. The microbiome adapts in response, not through disruption, but through rebalancing. Certain populations expand slightly, others recede, yet overall function remains steady. The system reflects the body's evolving needs without losing continuity. Throughout adulthood, the microbiome tends toward its most stable configuration. Daily rhythms reinforce predictable activity. Dietary patterns shape microbial efficiency. Immune regulation becomes well calibrated, allowing coexistence with minimal friction. Diversity remains present, supporting resilience and adaptability. This stage is not static, but it is reliable, capable of absorbing gradual change without loss of function. Life events introduce variation. Changes in environment, routine, or diet gently influence microbial communities. Travel, seasonal shifts, and evolving habits leave subtle impressions. The microbiome responds gradually, adjusting its balance while preserving essential roles. This adaptability allows digestion and regulation to remain effective across varied circumstances. As adulthood progresses into later years, physiological changes begin to emerge. Digestive movement may slow slightly, absorption patterns may adjust, and immune regulation may shift toward caution. The microbiome adapts accordingly. Populations reorganize to suit new conditions, often becoming less diverse, but still functionally stable. Reduced diversity in later life does not necessarily indicate dysfunction. Instead, it reflects a narrowing of environmental exposure and dietary variety. Core microbial functions persist, supported by species well adapted to long-term coexistence. Stability becomes more important than exploration, and the system prioritizes reliability over expansion. Throughout aging, the microbiome continues to interact closely with the immune system. Signals exchanged at the gut boundary help maintain tolerance and prevent unnecessary inflammation. This ongoing dialogue supports comfort and balance, even as physiological reserves change. The microbiome remains a quiet, stabilizing force. Importantly, the microbiome does not age independently of the body. It reflects the conditions provided, adapting to changes in digestion, movement, and metabolism. This interdependence ensures alignment between microbial activity and host capacity. Neither system advances alone. They continue together, adjusting to time. Sleep remains a central influence across the lifespan. During rest, microbial activity aligns with maintenance rather than growth. This alignment supports tissue renewal and metabolic stability. Even in later life, sleep provides a context for regulation, allowing the microbiome to support balance without strain. Across all stages, the microbiome demonstrates continuity rather than decline. Its form changes, but its purpose remains consistent. It continues to assist digestion, support immune regulation, and contribute to metabolic balance. These roles do not disappear with age. They adjust to new conditions. Understanding the microbiome across the lifespan encourages patience with change. Variation is not failure, but adaptation. The system does not seek youth or novelty, only balance. It reflects the body it inhabits, responding to time with flexibility rather than resistance. As you rest now, your microbiome reflects the history of your life. It carries traces of early exposure, long-standing habits, and recent routines. Yet it is not fixed in the past. It continues to respond to the present, adjusting gently to current conditions. Within you, this lifelong companion remains active, steady, and adaptable. It has supported you through countless days and nights, through change and routine alike. As sleep deepens, it continues its quiet work, aligned with the rhythm of rest. 18 Modern life and microbial simplicity. As human life has gradually changed over generations, so too has the environment in which the microbiome exists. These changes did not happen suddenly, nor were they guided by a single intention. They emerged from shifts in living conditions, food availability, sanitation, and daily routine. Within the body, microbial communities responded in the same way they always have, by adapting quietly to the conditions provided. Modern life is often characterized by increased predictability. Food arrives regularly and is prepared in consistent ways. Living spaces are controlled for temperature and cleanliness. Water is treated, surfaces are cleaned, and exposure to the natural environment is often limited. In earlier human environments, daily life involved frequent contact with soil, plants, animals, and diverse sources of microorganisms. This exposure introduced a wide range of microbial signals into the body, many of which passed through without lasting effect, while others contributed to microbial diversity. In more controlled environments, exposure is narrower. The microbiome responds by becoming simpler, not weaker, but more specialized. Simplicity in this context does not mean absence. It refers to a reduction in diversity, with microbial populations reflecting a smaller range of environmental inputs. Essential functions continue, supported by organisms well adapted to stable conditions. The system shifts from exploration toward maintenance, prioritizing reliability over variety. Diet plays a central role in this transition. Foods prepared in consistent ways introduce predictable substrates into the gut. Microbes adjust accordingly, refining metabolic pathways that match what is most often available. Over time, this specialization supports efficient digestion under stable conditions, even as diversity narrows. Sanitation and hygiene further influence microbial exposure. By limiting contact with environmental microbes, modern living reduces the number of new organisms entering the system. This reduction does not eliminate microbial life, but it does influence which species are most common. The microbiome becomes shaped more by internal conditions than by external influx. Medical practices also contribute to this environment. Interventions designed to protect health alter microbial conditions temporarily, and the microbiome responds by reorganizing itself afterward. Recovery unfolds gradually, guided by remaining diversity and functional redundancy. The system does not return to an earlier state exactly, but it reestablishes balance in a form suited to current conditions. Importantly, the microbiome does not evaluate modern life as beneficial or harmful. It responds only to availability and constraint. Reduced exposure leads to reduced diversity. Increased predictability leads to specialization. These outcomes reflect ecological principles rather than judgment. Within the body, immune regulation adapts alongside microbial changes. Exposure patterns shape immune expectations, influencing how tolerance and vigilance are balanced. In stable environments, immune systems often become finely tuned to familiar signals. The digestive tract continues to provide a supportive habitat. Temperature, moisture, and nutrient flow remain consistent, allowing microbes to persist even as diversity shifts. The body's internal environment remains one of the most stable ecosystems humans experience, regardless of external change. Rest and sleep play a stabilizing role in this context. During sleep, the absence of new inputs allows the system to integrate ongoing conditions. Microbial activity continues quietly, processing available substrates and supporting maintenance. This regular pause reinforces equilibrium. Simplicity does not imply fragility. A less diverse ecosystem can still be resilient if core functions are preserved and redundancy remains. The microbiome retains its capacity to adapt gradually when conditions change. It remains responsive rather than rigid. Understanding modern microbial simplicity as an adaptation rather than a loss encourages a neutral perspective. The system reflects the world it inhabits. As environments become more controlled, internal ecosystems adjust accordingly. This adjustment is neither sudden nor absolute. It unfolds over time. Within you, the microbiome mirrors your environment and habits. It has learned to operate within the conditions of your daily life, responding to routine rather than unpredictability. This learning supports continuity, allowing digestion and regulation to proceed smoothly. As you rest, this adapted system continues its work. Microbes metabolize what is available. Populations remain stable. Balance is maintained without effort or awareness. The system does not seek change, but it remains capable of it. Modern life has shaped the microbiome through consistency rather than conflict. The result is an internal ecosystem oriented toward steadiness. As sleep deepens, this steadiness persists, sustaining balance quietly through the night. 19. Integration. Digestion as continuous balance. When all the elements of digestion are viewed together, a single theme becomes clear. Balance maintained through continuity. Digestion is not a series of isolated steps, nor is it a process that begins and ends sharply. It is an ongoing flow, shaped by cooperation between human cells, microbial partners, neural signals, immune regulation, and daily rhythms. Each component contributes quietly, without demanding attention, sustaining the body through steady integration rather than force. From the moment food enters the body, digestion becomes a shared endeavor. Mechanical movement, chemical breakdown, microbial fermentation, and nutrient absorption unfold in sequence, yet overlap in time. No single stage operates alone. Instead, each prepares conditions for the next, forming a chain of support that extends across hours and even days. This continuity ensures that nourishment is extracted gradually and reliably. The microbiome plays a central role in this integration. It bridges gaps left by human digestion, completing tasks that enzymes alone cannot perform. Through fermentation and metabolic exchange, microbes transform complex substances into accessible energy and signaling molecules. These contributions do not stand apart from digestion. They are woven into its fabric, extending its reach and smoothing its pace. The intestinal lining serves as the meeting point where this integration becomes tangible. Here, nutrients pass inward, microbial signals are interpreted, and immune regulation remains attentive yet calm. This living boundary coordinates exchange without confusion, allowing interaction without intrusion. Its constant renewal ensures that balance is preserved despite continuous exposure and movement. Neural communication further integrates digestion with the rest of the body. Signals traveling between the gut and nervous system align digestive activity with overall physiological state. During activity, digestion supports energy availability. During rest, it shifts toward maintenance. This alignment ensures that digestion complements rather than competes with other bodily demands. Immune regulation adds another layer of integration. Rather than reacting reflexively, the immune system interprets microbial signals within context, maintaining tolerance where appropriate and readiness where necessary. This measured response prevents disruption and preserves cooperation. Daily rhythms provide the temporal structure within which all of this occurs. Eating, resting, and sleeping create predictable patterns that guide digestive activity. Microbial populations adapt to these rhythms, synchronizing metabolism with nutrient availability. Over time, this synchronization enhances efficiency and stability, reinforcing balance through repetition. Change is absorbed rather than resisted. Variation in diet, routine, or environment introduces new conditions, and the system responds by adjusting gradually. Diversity within the microbiome supports this adaptability, ensuring that essential functions persist even as populations shift. Stability emerges not from rigidity, but from flexibility supported by redundancy. Throughout this process, digestion remains largely invisible to awareness. Its success lies in its ability to function without drawing attention. When balance is maintained, there is no sensation of effort or strain. The body simply continues, supported by countless interactions unfolding beneath perception. Rest reveals the continuity of digestion most clearly. During sleep, when external input pauses, internal processes continue their steady work. Food consumed earlier is still being processed. Microbial fermentation releases energy slowly. Nutrients are absorbed and distributed. Cells repair and renew. The absence of conscious activity allows these integrated processes to proceed without interruption. This ongoing balance reflects evolutionary refinement. Over generations, systems that favored cooperation and gradual processing proved more resilient than those dependent on speed or intensity. Digestion evolved to be thorough rather than rapid, integrated rather than compartmentalized. The microbiome became an essential partner in this approach, extending capability without increasing strain. Understanding digestion as continuous balance can bring a sense of ease. There is no need to manage or oversee each step. The body does not require constant correction. It relies instead on systems shaped to adjust automatically, guided by feedback rather than command. Within you, this integration is happening now. Even as you rest, muscles contract gently, microbes metabolize steadily, signals travel quietly, and barriers regulate exchange. None of these actions require awareness. They proceed according to patterns established long ago, refined through daily repetition. Balance does not mean absence of activity. It means appropriate activity distributed across time. Digestion exemplifies this principle, spreading work so that no single moment bears the full burden. Energy is released gradually, maintenance is ongoing, and recovery is built into the process. As sleep deepens, this continuous balance remains intact. The body does not pause its essential functions. It simply shifts emphasis, allowing integration to take precedence over response. This shift supports restoration, preparing the system for the next cycle of activity. Seeing digestion in this way highlights the quiet intelligence of biological systems. They do not rush toward completion. They maintain equilibrium through cooperation, timing, and adaptability. The microbiome, the gut, within the stillness of rest, this integration sustains you. Nothing needs to be accelerated. Nothing needs to be resolved. The body continues as it was designed to do, guided by continuity rather than urgency. As the night progresses, digestion remains part of the gentle hum of life within. Balance is preserved, energy flows steadily, and internal systems remain aligned. In this quiet integration, the body finds its strength, carrying you calmly through the hours of sleep. 20. A slow closing reflection. As the body settles more deeply into rest, it can be comforting to remember that nothing within you has been left unfinished. Digestion continues in its own time, neither hurried nor delayed, guided by rhythms that do not require attention. The work of transformation, absorption, and balance proceeds quietly, as it has throughout your life, sustained by cooperation rather than effort. Within you, the human body and the microbial world remain gently intertwined. Human cells maintain structure and direction, while microbial partners contribute patience and persistence. Together, they form a living system that does not pause when awareness fades. Even in sleep, this system continues, carrying out the small, steady tasks that sustain life from moment to moment. The microbiome does not seek recognition. It does not announce its presence or demand care. It responds simply to the environment it inhabits, adjusting its activity to what is available and what is needed. In this responsiveness lies its strength. It supports digestion not through force, but through quiet alignment with the body's needs. The digestive tract itself remains calm and attentive. Muscles contract gently, guiding contents forward. Cells absorb nutrients as they become available. Barriers regulate exchange with care and precision. None of this requires conscious direction. The system knows how to proceed, having learned its rhythms over countless repetitions. Throughout the night, fermentation continues at a measured pace. Energy is released slowly, supporting maintenance rather than activity. Byproducts nourish the intestinal lining, reinforcing its integrity. Signals travel softly along nerves, maintaining communication without disturbance. Immune regulation remains present but untroubled, preserving balance through restraint. All of these processes are expressions of continuity. They do not begin anew each day, nor do they end with sleep. They flow onward, adapting gently to changing conditions while preserving their essential purpose. This continuity allows the body to rest without fear of neglect. It can be reassuring to recognize that digestion is not something you must manage. It is a living process that unfolds according to its own intelligence, shaped by time and cooperation. The body does not ask for vigilance, only for the opportunity to continue as it knows how. As sleep deepens, external concerns recede, the mind grows quieter, and attention turns inward without effort. In this quiet, the internal world becomes more prominent, though still unseen. Cells renew themselves, microbes continue their steady work. Balance is preserved, not by control, but by trust in the systems that sustain you. The microbiome reflects this trust. It has adapted to your habits, your environment, and your rhythms. It does not expect perfection, only consistency. It responds to what is present, making use of available resources and adjusting when conditions change. This adaptability allows it to support you through varied experiences without losing its grounding. Over the course of this exploration, digestion has revealed itself not as a single pathway, but as an integrated landscape. Each region of the gut contributes in its own way. Each microbial community plays a role suited to its environment. Together, they form an ecosystem that exists within you, shaped by cooperation rather than command. This internal ecosystem mirrors the broader patterns of life. It seeks balance, achieved through countless small interactions that accumulate over time. As you rest now, this balance remains intact. The food you have eaten is still being transformed. The energy it contains is being released gradually. Nutrients are finding their way to the cells that need them. Waste is being prepared for release. Nothing is rushed. Nothing is left incomplete. Sleep offers a pause from outward engagement, but it is not an absence of activity. It is a shift in emphasis, allowing maintenance and restoration to take precedence. Digestion fits naturally into this phase, continuing its work without interference. The microbiome remains active, yet aligned with the calm of rest. In this stillness, the body's wisdom becomes apparent. It does not depend on constant oversight. It does not require reminders. It has been shaped by evolution to care for itself, drawing on partnerships that extend beyond human cells alone. This shared care allows life to continue smoothly, even when the mind is at ease. There is nothing you need to do now. The systems within you are already at work, sustaining balance gently and reliably. Each breath supports this work. Each moment of rest allows it to proceed more fully. Sleep becomes not a withdrawal, but a continuation of life in a quieter form. As the night progresses, the boundaries between one process and another soften. Digestion, immunity, neural regulation, and microbial activity blend into a single, steady flow. This flow carries you through the hours of darkness, preparing the body for the return of light and activity. In recognizing this continuity, there is room for ease. The microbiome remains a faithful participant, contributing quietly, without demand. As you drift further into sleep, let this understanding settle gently. Within you, an entire world continues its calm, cooperative work. Nothing is missing. Nothing is urgent. Balance is being maintained moment by moment.